Hi there, I'm Mrs. Gay, and I'm starting on page 52, Chapter 4, Section 1. This section is about microscopes, and quite honestly, all you really need to do is be familiar with the fact that there are different types of microscopes, and we will work largely with a light microscope. And so think about the onion root cell. You're never going to get asked to name the parts of a microscope. You're never going to get asked how to focus a microscope. But you should be aware that many of the things that we study in biology are smaller than we can see with the naked eye, and that the microscope is a valuable tool to help us see things that we normally couldn't see. So we have the light microscope, we have an electron microscope, scanning and transmission. So a scanning electron microscope shows us the surface of things, and while a transmission microscope actually puts energy through the object and we can actually see inside. Okay, that's plenty to know. The one really important thing in this whole section is at the first paragraph in the second column of page 52. Okay, and it's probably not a huge deal, but Robert Hooke discovered cells. Cells, and this is actually total trivia, cells were not, don't think prison cells, but cells were small rectangular quarters that were occupied by monks in monasteries. And that's what they reminded him of. And so that's why he called them cells. Okay? And so that's a little bit different. But the one piece that's in here listed is cell theory. Okay? And we'll go back and revisit cell theory in another section in this book too. But the big points about cell theory is that the cell is the basic unit of all living things. That's sort of one facet of the cell theory. Okay? The second big idea about the cell theory is that all cells come from pre-existing cells. Okay? And we'll see in a later section that these ideas came first from Virchow and then later from Schleiden and Schwann.
Do you want...
I know, they were both German.
Do you want the name, like, would a question on the test be, what are the names of the...
No, they would not ask you who discovered, who were the major contributors to cell theory and have a whole bunch of names. But they might mention Schleiden and Schwann in association with cell theory. And so I mention it because I want you to, if you see it, be, oh, right. You may not be able to pull it out of your hat, but if you saw it, you wouldn't freak out. Like, oh, my God, I have no idea who these people are or what they did, when really it's not that big a deal. Okay?
So I would say, like, the theory formed by
Schleiden and Schwann. Or if you think about it, okay, so if you think about it on the free response question for the cell membrane essay that I asked you to write, and it was talking about the fluid mosaic model, and it was talking about the Singer model versus the Danielson model, or Danieli, I can't even remember who the other person was, model. If you knew who had come up with that idea and you listed it, you might get a point. But there were plenty of other ways to get your 10 as well. So if you're good with names and you like sort of historical things, great, remember the names. But it's not going to hurt you or necessarily help you a whole lot either way. But I tell you them mostly so that you would be familiar with them and not be surprised if you saw them. Does that make sense? Yeah. So I would not, I would look over this section, but I would not spend an extraordinarily large amount of time on that section. So we're going to move on to section 4.2, which is a big deal. Okay? Most cells are microscopic, and if you look at figure 4.2a on page 54, you can see what we can see with the unaided eye. You can see that overlaps with what we can see with the light microscope, which also overlaps with what we can see with an electron microscope. Okay, so you should have a sense of size. Okay, notice this though, that's interesting. We'll come back to this later. A chicken egg. Notice how big it is. Ostrich eggs, emu eggs, even bigger. Frog eggs, smaller. But notice most plant and animal cells, the nucleus is actually bigger than most bacteria. A mitochondrion is smaller than most bacteria, but not a lot smaller. And that's a preview of endosymbiotic hypothesis, how we got from prokaryotic to eukaryotic. Notice that viruses are even smaller. Ribosomes are even smaller than a virus. And then we get down to the macromolecules and smaller. So just having a small sense of the scale of things. The other really important idea in this chapter is the idea of surface area to volume. It's all about surface area. Okay? And the reason that it's all about surface area, as things get bigger, the surface area only increases as the square, where volume increases as a cube. Okay? And at the top in figure 4.2b, they give you a really nice illustration of what happens where the volume remains the same, but they break it up into smaller cubes and we drastically increase the surface area. We find that happening all the time. Think about your brain, think about your intestines, think about your lungs. Everything is highly folded and convoluted to give greater surface area.
And what's the benefit of having that?
Well, the benefit of having a large surface area, think about exchange of nutrients. And think about your osmosis diffusion lab with those agar blocks. They started out pink, we put them in hydrochloric acid, they turned clear. Which cube took the longest time to get clear? The big one. Now think about this. I could have had a cube that had the same volume as the biggest cube, but what if I would have made it flat and long and thin? It would have taken way less time because we would have exposed much more surface area than by keeping it in its nice sort of handy cube shape. Does that make sense? And so surface area to volume matters at a cellular level because we have to get waste out and we have to get nutrients in. And so if you think about nutrients diffusing into the cell, that really big cell that stayed pink in the center, nutrients never got into the center of the cell. Waste in the center of the cell still would be inside the cell. And that would end badly for life. So it's about exchange with the environment.
Are there like cells that have like greater, like just an example of an actual cell that has better surface area to volume than other cells do? Like
Sure. Think about a nerve cell that would be really long and thin. That's going to have a much greater surface area than, say, a cuboidal cell.
So just overall thickness.
So, right, so thinness and stretching it out. Think about a roll of aluminum foil. You could take a roll of aluminum foil and you could wad it up into a cube. Okay, and you'd have a cube of aluminum versus rolling it out super thin. It's going to have much more surface area all rolled out thin. But what do we have to do with the aluminum foil all rolled out thin? That would be really tough to store unrolled.
Yeah.
So what do? We fold it up, we roll it up, we compress it. And so if you think about the internal structure of much of you, you're folded and convoluted. Okay. So I'm going to stop there. We'll start with Memo 2, looking at Section 4.3. There we go. Hi there, I'm Mrs. Gay. We're now on Section 4.5. Okay. This section is a preview of coming attractions, even though we did it in reverse order. Okay, this is looking at the structure of membranes, and when we look at this little section, it seems really obvious. And in fact, we saw this little phospholipid diagram, the gray and the yellow diagram, and we also saw 4.5b diagram way back when we were studying lipids. Both of those diagrams appeared in Chapter 3. So, phospholipid bilayer, Okay, made out of phospholipids, 2 fatty acid tails, which are hydrophobic, nonpolar. They could be saturated, they could be unsaturated, or polyunsaturated for that matter, right? And instead of the 3rd fatty acid, there's a phosphate group, which is hydrophilic because it's polar. So the molecule is said to be amphipathic. Ampha meaning both. So it has both polar and nonpolar characteristics. That word is actually not in your book. Okay? And so you see that we have the phospholipid bilayers so that the hydrophobic regions can be tucked away together on the inside. The hydrophilic regions are pointing away to the outside. And it's a fluid mosaic model because those phospholipids can move around between each other, and then we have proteins embedded in the membrane. Some of those proteins are just surface proteins. Some of those proteins are just stuck on the inside of the cell membrane, and other proteins actually pass all the way through. So, I've always wanted to do this. You know how Nilsson takes everybody down to the pool for the Cardboard Classic? I would really like to take everyone down to the pool to make a phospholipid bilayer model. And so what I would do, and my problem is I don't have enough ping pong balls. So what I would do would take barrels and barrels and barrels of ping pong balls, and I would just, we could only see the top part of the fluid mosaic membrane, but I would just cover the whole surface of the swimming pool with ping pong balls. And can you imagine if there were little waves, the ping pong balls would sort of move, right? And then I'd lay a raft, a small raft, on top of the ping pong balls. And that would represent a protein on the external part of the membrane. And then I could put something that would float under the ping pong balls, which would represent a protein on the inside of the phospholipid bilayer. And then I'd take a big old inner tube and throw it in. And that would be a protein that goes all the way through. And it could be like an aquaporin, a pore. And then you could jump through the inner tube into the inside and outside. Yeah, so water could pass through it. So it could be like a channel protein. Ooh. A protein that goes all the way through the phospholipid bilayer is called an integral protein or an integrin. Okay.
This is all later too.
This is all, yes, integral or integrin. This is all later as well in the first part of chapter 5. Okay.
Wait, an integrin goes all the way through,
Is-
but isn't a channel.
It could be a channel, it may not be a channel. Okay. The proteins actually do many things. Okay. Now, so we move on then to start looking, but most of a eukaryotic cell is sort of membrane bound. So the first thing we have is that we're subdividing the cell into organelles that manufacture things and break things down. Oh gosh, anabolic and catabolic again. Making things and breaking things down. Dehydration synthesis, hydrolysis. Okay? And so, if we want to make stuff, we need directions. Okay? I am knitting Norwegian sweaters. I'm dead serious. Last weekend, I retaught myself how to knit. I used to know a long time ago. But do you think I could have figured out how to knit all by myself? Maybe eventually. But really, what I needed were directions that told me how to cast on, get the yarn on the needle in the first place, and what the difference between knitting and purling and how many stitches and how to knit with 2 different colors, right? And then I needed directions. If I'm going to knit a Norwegian sweater, I need really cool Norwegian patterns in my Norwegian sweater, right? And so I needed directions. So, no directions? How do you think my mitten would look?
Probably wouldn't.
Not so good, and it probably wouldn't be very functional. So directions are key. The directions for making everything found in the nucleus. Okay? And so some of the key words. that they have in this section. The nucleus is surrounded by a nuclear envelope. Notice it's a double layer. Okay, so there's two membranes that form the nuclear envelope. There are nuclear pores. Now notice that in your textbook, they look like the inner tube floating in the ping pong balls, right? Remember the cellular landscape? Is it really a hole? No. It's more like sort of a loose mesh network that you have to get through, right? I think it was green in the cellular landscape, if I remember correctly, the nuclear pore, because it wasn't actually a hole.
Yeah, it was green.
Yep. And so you can look at it back in the classroom if you need to remember, right? Inside the nucleus is the DNA. There are many, many, many names for DNA. If the DNA is all unwound, it's called chromatin.
Unwound DNA.
Unwound DNA. So think about a spool of thread. Chromatin. So if I took my spool of thread and I totally unwound it, I'd have this giant pile of loose thread. That would be chromatin. If I wind the thread back up on the spool, it's going to be more protected, it's less likely to get broken. At that point, I call it a chromosome. Preview of coming attractions, if I've made a copy of that spool of thread, my two spools of threads are called sister chromatids. So we have chromatin, chromosome, chromatid. Okay, does that make sense? So all of these words are referring to the DNA in sort of different packaging. We also have the nucleolus. Okay, that's where we synthesize ribosomal RNA. And ribosomal RNA and proteins make up the ribosomes. So sometimes the nucleolus is sometimes called the site of ribosome synthesis. It doesn't actually like totally make the ribosomes there, but it actually produces the ribosomal RNA.
And that's all it does.
Okay?
Is the nucleolus inside the nucleus?
Yes, the nucleolus is inside the nucleus.
And its main function is synthesizing
Ribosomal RNA. Okay, so we bring proteins. Proteins actually get made out in the cytoplasm. They get brought into the nucleus through the nuclear pore, and then they get assembled with RNA, ribosomal RNA, and we make the subunits of the ribosome and ship them back out. Okay? So, if we talk about the ribosomes, which get initially sort of built in the nucleolus, ribosomes are the sites of protein synthesis. There is a large subunit and a small subunit. If ribosomes can be floating around in the cytoplasm, in which case they are free ribosomes. Other ribosomes are bound to the endoplasmic reticulum. Okay? So we can find them in two places. Most of the proteins that are made on free ribosomes stay in the cytoplasm, and then they function inside the cell. Whereas often, proteins that are made on the endoplasmic reticulum get packaged and shipped to other places outside the cell.
The free ones don't.
But the free ones generally produce proteins that stay inside the cell. Okay? Now, in section, and I'm going to stop there because then we're going to continue on with the endomembrane system in the next section. Hi there, I'm Mrs. Gay. And we are moving on to energy converting organelles on page 63, section 414. This gives you a brief introduction to the mitochondria. This goes back to things that you would have known in 10th grade. The outer membrane, the inner membrane space, the cristae, the matrix. We will go into the mitochondria in much more detail when we study cellular respiration. Okay? Same thing with the chloroplasts. They give you an introduction to chloroplasts, but again, we'll dedicate much more time to the chloroplasts when we study the process of photosynthesis. They're giving you an introduction to the idea that mitochondria and chloroplast evolved by the endosymbiotic theory, put forth by Lynn Margulis, a woman, okay, who worked at Harvard, but has since passed away. Very brilliant woman. But we'll talk again more about endosymbiosis in a future chapter. The last little bit of this chapter is looking at internal and external support, the cell surface and the cytoskeleton. So the cytoskeleton provides the cell with structure, it allows it to move, it holds the organelles in place. Really think about the inner life of a cell video and think about the cytoskeleton forming and breaking apart. Think about it forming those microfilaments and microtubules that sort of kept the mitochondria sort of aligned and in place so things just aren't sort of slopping around. And so the cytoskeleton is composed of microfilaments, which are made out of actin, intermediate filaments, and microtubules, which are made out of tubulin. Okay? Microtubules are made in an organelle called a centrosome. Inside the centrosome are two orthogonal, which means perpendicular, centrioles.
This is the microtubule.
Wait, micro.
Okay, so okay, we have a center called a centrosome. Inside, and so the centrosome makes microtubules.
That's where the spindle comes from.
Mm-hmm. Excellent.
Okay.
And if you're an animal cell, inside the centrosome, you also have two orthogonal, perpendicular, centrioles. And yes, that's what constructs the spindle fibers in cell division.
Just for animal is the centriole.
Centrioles are only found in
We're not
animals.
sure what the function of centrioles is.
Right.
Is that worth a Nobel Prize?
Could be. It could be.
It's like going for it.
This year's Nobel Prize was given for G-coupled protein receptors,
Like in that video.
like in the video. And we'll learn more about G-coupled protein receptors because they're just so cool and amazing. And so it's all about cell signaling. Okay. The other Nobel Prize that's of interest to us, the physiology and medicine one, was given for stem cells.
Are they getting closer?
They're getting closer all the time, particularly in countries where research is not regulated, like it is in here.
I know this is off topic, but did you see the 60 Minutes?
So now we're going to focus in section 418. We're going to focus on cilia and flagella, which are made out of microtubules. And probably the good thing to know here is that they're organized. We have several microtubules, and they're organized in this 9+2 arrangement. 9 pairs around the outside, 2 in the center. And of course, this is only scratching the surface. There is much more we could learn about this. And they use little dianin arms to sort of bend and pull, which is what causes the cilia and the flagella to bend and wave, which is how we get motion from, for example, the cilia that line our respiratory system to the flagella that move sperm along or move bacteria along.
So what gives it its wavy?
Little proteins, it's made out of tubulin proteins, and so the little tubulins actually have like little dianin arms. And so if you can imagine if there was a rope over my head, if I reached up and grabbed the rope and moved it, I could reach up and grab the rope and move it, and I could make it move.
Mm-hmm.
And so if that happened on the top, it could make it curve up. If that happened on the bottom, it could make it turn down. And so the flagella are attached to the cell with a basal body. Okay? The basal body has even more microtubules. They're arranged in triplets, and these details are beyond the scope of the class, so don't get all hung up about, triplets versus pairs and all that stuff. But there's also tons of mitochondria there to provide ATP to power that flagella in its bending process.
And it's at the base, like right
Right.
between the, like on the sperm cells, like right at the base of
Right. Okay, exactly where it's attached. And then they give you a little connection that sperm motility could be genetic or it could be environmental if they're not producing the right sort of tubulin structures. The flagella is not going to be very mobile.
Do flagella and cilia have different amounts of dyning proteins because they move differently?
I don't know if they have different amounts of proteins. They're different lengths. And so their overall movement's going to be different. Now, in a prokaryote, their flagella, instead of going up and down like this, they actually spin around like a propeller. Yeah, which is a whole other story. Okay, and so how cilia and flagella actually move in detail is well beyond the scope of this class. That would be a class called cell biology, where we're really looking at this stuff for a whole semester. Can you imagine the stories they could tell? I know. Now,
Kelly's excited.
yeah.
Time
So-
check, it's 8:08.
Perfect, I know. So we better get rolling. Thank you.
Sure.
We also have the extracellular matrix because cells have to stick to other cells. And then at the end of this chapter, we're looking at 3 types of cell junctions.
Wait, what does the extracellular matrix do?
The extracellular matrix holds cells together. Remember, cells need to stick together, anchorage dependent,
Yeah.
and contact, and so they help hold cells together.
And integrins do that, like actually.
Mhm. And if you look at this picture on page 67, 420, you can see that there's collagen, there's glycoproteins, there's the integrin proteins, okay, with long polysaccharides. And so there's all this stuff sort of hanging out in cells. I know.
So like the integrin are actually going like through the membrane and like reaching on both sides.
And they reach out on both sides. Yes, that's important. And then the last thing that you should review is just the differences that connect cells, the differences between animal tissues and plant cells. So in animals, we have tight junctions that basically compress them tightly together, and it sort of makes a seal, which keeps things from leaking, which is good. It would be awkward and embarrassing if you leaked. Okay? Yeah.
The analogy of the swimming pool, that was
Right. Yes. Yes. So remember the swimming pool. We have anchoring junctions that work like rivets that really sort of keep you secure. And I think we talked about this when we were talking about like the cupboards being anchored to the walls. And then we have gap junctions because cells have to communicate. And so we have little gaps. They allow small little molecules to pass through and signal each other. In cells, we have cell walls, and so that makes things a little more tricky. So, in plant cells, so we have plasmodesmata. I love that word, plasmodesmata.
It kind of sounds like calming a little bit.
I know, doesn't it? Some sort of Zen, plasmodesmata. And if you notice in the diagram, actually, the cytoplasm can actually flow from one cell to another through the plasmodesmata.
So all plant cells are like kind of one big cell.
Yes. Uh-huh.
Oh, okay.
Yeah.
So animal cells have the tight junctions, but do both have anchoring junctions? Or just animals?
Well, animal cells have tight anchoring and gap.
They have all, but like do
And so
anchoring.
plant cells, actually their cell walls sort of get knitted together with lignin and other cellulose kind of proteins.
So which one has integers?
Integrin proteins?
Yeah.
They both do because we're talking about the cell membrane, not the cell wall. Integrin proteins, okay, integrin proteins span the entire membrane.
So they're the ones that do like transport
Mm-hmm.
and stuff like that.
Right. And then the last thing I want to say, and then I'll stop the recording and you can ask any final questions, but then we're going to really start moving on, is on page 69, that last section gives you that really nice table and sort of sums it up, the whole thing. And so that would be a really nice thing to look at as well. So, most organelles are basically just made of membranes. And so we call it the endomembrane system. I'm looking at section 4.8 on page 59 in your book. Okay, so most of the organelles in the cell are made out of membranes. And because they're made out of membranes, they're still made out of this phospholipid bilayer with proteins embedded and on both sides and da-da-da. Because it's all membranes, they can break off. They can fuse. Bits of one organelle can become bits of another organelle. We saw that with the inner life of a cell, that video, how membranes could bleb off and become vesicles. Those vesicles could fuse into the Golgi apparatus. Okay, so a little sac surrounded by membrane is just called a vesicle.
Sack of what?
It could be lots of things. It could be a sac of digestive enzymes. It could be a sac of food. It could be a sac of protein products that we want to dump outside the cell. So any sort of little sacs made of membranes that hold stuff are called vesicles. Sort of a generic name. Okay?
And are organelles technically vesicles as well because they're membrane?
They are, but they tend to be a little larger than a vesicle. And so, for example, endoplasmic reticulum are these long tube-like structures of membranes. Okay? And so...
What's the difference between a vesicle and a vacuole? Is it the same purpose?
Yes.
Okay.
A vacuole is a large vesicle used for storage.
Okay.
Okay? So, endoplasmic means within the cytoplasm. Reticulum means little net. And so, a reticulated giraffe has a net-like pattern of skin coloration. All right? And so, we have two types of endoplasmic reticulum. We have smooth, we have rough. In section 4.9, on page 60, we're really looking at endoplasmic reticulum as a biosynthetic factory. It makes tons of stuff. Smooth endoplasmic reticulum makes more lipids. Well, golly gee. Oils, phospholipids, steroids. So if we want to make more membrane, there we go. Okay. Liver. Our liver has huge amounts of smooth endoplasmic reticulum because we have to use it as our detox center. Okay. Smooth endoplasmic reticulum also stores calcium, which is important in muscle function. It's also stores calcium. It's also really important in nerve function. Calcium's important in both. Okay. Rough ER. The main function of rough endoplasmic reticulum is to make more membranes. So we insert phospholipids that are made by the enzymes, and we insert them into the membrane so the membrane can grow. And if you think about it, that's really important because the endoplasmic reticulum is going to pinch things off, so it's continually losing membrane. So it needs to make more. Yeah.
But so the smooth ER makes lipids, which in turn makes membranes, but the rough ER makes membranes.
Right, so the rough ER is going to sort of assemble them because it's also making the proteins that are going to be embedded in the membrane.
So the rough ER assembles what was made in the smooth ER.
The rough ER is like the one where there's kind of like it enters one side and comes out the other in that central like whole
But
thing.
that was the
Isn't that the
Golgi
Golgi?
body.
That's the Golgi apparatus, I think.
That is what it is.
Yes, so the rough endoplasmic reticulum is like a flat tube. It has ribosomes embedded in it. Okay, and so if you look at figure 4.9b, notice that the ribosome has docked on the endoplasmic reticulum. As it's synthesizing the amino acid chain, it synthesizes it into the center or the lumen of the endoplasmic reticulum. It gets folded up. We're adding some sugar chains to the outside, so we're glycolating it. So now it's a glycoprotein. And notice it moves towards the end, and this little transport vesicle buds off, pinches off. And now we have a little transport vesicle. The protein may not be fully folded, and so we may take that little transport vesicle. Remember our little motor proteins marching along the cytoskeleton. They're little fuzzy slippers. Okay. They travel to the Golgi apparatus. That little transport vesicle holding the unfinished protein is going to fuse, because it's a membrane, it can fuse with the membrane of the Golgi apparatus. And now our little unfinished protein is on the inside of the Golgi apparatus, where it can continue to be modified. We could snip little pieces off, we can add other little pieces to it and make a quaternary level structure. We have other proteins that are called chaperonin proteins, which are sort of like barrels with lids. And so that protein, that unfinished protein, gets stuffed into another protein barrel. And that protein barrel squishes it into shape to give it its final tertiary quaternary level structure. Who knew, right? And so the Golgi apparatus sort of finishes, sorts. It's sometimes called the packaging center of the cell. And on the far side, so can you imagine this? The Golgi apparatus is not static at all. It's continually forming and fusing. And so if you think about time-lapse photography, up here in the ceiling, vesicles are coming and they're fusing together and they're making the top layer of the Golgi apparatus. It's going to get pushed down. That layer is going to get pushed down as more vesicles come in and fuse. And as it gets pushed down, pushed down, pushed down, those proteins inside get polished and packaged and folded up into their perfect shape. And so by the time that they actually get down towards the bottom, they're ready for, remember those clathrin proteins, those sort of X-shaped proteins, came in and pinched off part of the Golgi apparatus down at the bottom. We can pinch off another vesicle.
The claw thing.
Yeah, like the claw that pinched off the end of the Golgi apparatus, and then it too can get transported along with the motor proteins out towards the edge of the cell, and that vesicle can fuse with the plasma membrane, and poof, the insides are on the outsides. Right? Inside you right this very second.
So weird.
It's so weird to think about it. Yes, go ahead.
With the glycoproteins, does that refer to anything with a carbohydrate? Is that like a carbohydrate and protein together?
Right. So often proteins get little flags put on them with different carbohydrates. So for example, your ABO blood group, your red blood cells, are flagged with different carbohydrates. And so I'm type A blood, and so I have a certain type of carbohydrate protein flag sticking out of the cell membrane that says A. If you have type B blood, you have a different glycoprotein sticking out of the plasma membrane that says B. If you have type AB blood, you have both. And if you have type O blood, you have no little glycoprotein flags. Yeah.
I have O.
Right. And so, and that will come into play when we talk about the immune system and we look at antibodies, because what sticks to what. And so glycoproteins are often used in cell recognition, okay, and cell identity. So, go ahead, Kelly. I'm sorry.
So I had a question on one of the these things.
Practice multiple choices. Yes.
There was a question that said, which organelle is involved in the catabolism of fatty acids and the detoxification of alcohol? And so the smoothie R, I said the smoothie R, and that wasn't right. So does the smoothie R function in the detoxification of alcohol, though? Because it said it broke down harmful substances, and it talked about how there's large quantities in the liver.
Yes. Well, so I would have thought, what did it say the correct choice was?
Peroxisome?
Yeah.
Ah, yes. Well, so we will actually look at peroxisomes.
This was 4.6223, and it's question 9.
Yes, it is. And so when we get, so there's a couple of little vesicles. So you can imagine a protein product being produced. Okay, and so yeah, you might think that it would be involved in detox. It turns out it actually is peroxisomes. And so if you look ahead to section 4.13 and you look at the second paragraph, a peroxisome is an organelle that is not part of the endomembrane system, but is involved in various metabolic functions, including the breakdown of fatty acids to be used as fuel and the detoxification of alcohol and other harmful substances. Yeah, it was a little sneaky. Okay?
So the smoothie R, it does also function
It does
to detox
detox other chemicals.
that. Okay.
Yeah. And so you can imagine that in your liver cells, you'd still have a fair amount of peroxisomes as well as smooth ER. Okay? Lysosomes. Lysosomes are special little vesicles that contain digestive enzymes. And that's really important because this endomembrane system sorts and separates. We want places where stuff is and we want places where stuff isn't, because you wouldn't want your digestive enzymes that you produced just to be hanging around inside your cell, because what would they do? Digest everything.
So they're like the little safety.
Right, and so a lysosome is like a little sac, a vesicle, that contains digestive enzymes. If that lysosome were to break, the cell would get digested from the inside out. So sometimes lysosomes are called suicide sacs. Okay? If a cell needs to get rid of itself, it pops the lysosomes. And the word for cell suicide, if you will, does anybody know what that word is? Apoptosis. Apoptosis.
What circumstances would a cell want that to happen?
Oh, well, stay tuned for chapter 8. If a cell starts dividing out of control, a cancer cell, an early forming cancer cell, apoptosis is a mechanism to eradicate and kill a faulty cell.
Is that what, like,
Is that what cancer?
yeah,
Chemo?
chemotherapy? Like,
Chemo just kills cells.
Chemo, yes, it actually targets, chemotherapy actually targets rapidly dividing cells. So think about this, tumor cells,
Your
cancer
nails.
cells are rapidly dividing. So why do you lose your hair?
because that's always.
Because it's those cells are rapidly dividing, and so it kills those cells.
Do your nails stop growing when you have chemo? I've always
Well, I thought they, yeah.
I
I
think,
think
I
they
think
get
so.
really brittle. And
yeah, they get really brittle, and I think it slows down and
Like they
hair
just stop
everywhere,
growing.
like on your legs and stuff too, not just
Right.
red.
Yes, yeah,
Like
all
eyebrows
hair.
and eyelashes.
Mm-hmm.
And everything.
Yep.
That suck if your nails fell out.
I don't think they actually
They don't.
fall
No,
out.
they don't.
They just stop growing.
so bad.
Yes.
So I wanted to ask this earlier when it was more on topic.
Oh, you know what? Can I stall your question for just one moment? Because I want to just finish the endomembrane system.
Well, we're recording.
And then, we'll pause for a few questions, and then we'll finish up with the energy organelles, and then we'll be halfway done.
Actually, continue.
Okay, so you can sort of see in figure 4.11b, you can see how a lysosome could fuse with a food vacuole if we didn't want to kill the whole cell. Okay, it's fusing with a food vacuole, and those digestive enzymes are digesting the macromolecules by hydrolysis down into individual monomers to be recycled then and used in building other parts of the cell. So we're continuing to recycle.
Is that what happens inside your stomach?
Um,
Or is there just a lot of enzymes in there?
it's just producing, it's not doing it at a cellular level, it's doing it as an organ level.
Okay.
And so, yes, it's dumping in digestive enzymes that are breaking up and breaking apart big chunks of food down into small macromolecules. But they may still be fairly large, like proteins. So we may be sending proteins down into our digestive system, and those proteins may get taken in. So, yeah, so we're going to break things down into individual monomers. At a large scale, that's our stomach. But if you're a single-celled organelle, and you phagocytize some food, or another little bacteria, or another little single-celled organism, you need to break those guys up. And so, in some ways, it acts like a cellular stomach. Okay? It also, you'll notice in the bottom picture, it's recycling a damaged mitochondria. Think about those guys. Those little powerhouses generating all that ATP, they're going to wear out.
So do damaged organelles, all damaged organelles get consumed by a vesicle, which can then be further broken down by lysosomes?
Well, so the damaged vesicle would just get engulfed by the lysosome.
No, if there's like a,
All right, so I'm sorry, back up. It would get enclosed by a vesicle,
yeah,
and then the vesicle fuses with the lysosome.
which would in turn break it down into
Right.
Okay.
And then what happens to that?
Material?
That
Isn't it secreted from the
It's recycled.
It could either be secreted from the cell, or since it's now just a loose amino acid or a pentose sugar, or it's just a single sugar, it can float around in the cell, in the cytoplasm, as a monomer until it needs to get assembled, because now it's like loose in the Lego bin, waiting
And
to be put together.
Is that when it's like released? Is it still a lysosome? Does
So we're chapter 5 now?
Hi there, I'm Mrs. Gay, and we are now on Chapter 5, Section 1, on page 74. And what I want you to do, we're going to take just a closer look at cell membranes, and if you look at the figure 5.1a, you can see the phospholipid bilayer. You see cholesterol molecules in there, right? All right. And so then you also see proteins. Some of those proteins are only on the outside, the one in the far left. Some of the proteins are only on the inside, see the one on the far right. Some of them are integrin proteins, which go all the way through. Okay. Did I mention when I said cholesterol, did I mention that cholesterol helps to stabilize the membrane and give it a little bit of structure? And then notice that we have a glycoprotein, and that's a protein with a carbohydrate hanging off the end, and that is used in cell-to-cell recognition. Okay. We also can have glycolipids, and notice here that what we have is a carbohydrate chain sticking out off of the end of the lipid. Glyco meaning sugar, so a glycolipid is a lipid with a sugar stuck out on the top of it. Okay?
And does that also do cell-to-cell?
Uh-huh. Yes. And so the glycoproteins and the glycolipids really function in cell-to-cell recognition.
So a glycolipid is just a lipid with a sugar chain?
Yep. Yep.
And both of them serve the purpose of flagging?
Flagging and recognizing, yes. Okay. Now, many of the proteins that are embedded in the membranes are enzymes, and they actually do jobs, and they work as a team. So imagine an assembly line. I'm enzyme 1, and I break something down. I hand my products to Kelly, who's a second enzyme, and the products of my reaction become the reactants and fit in your active site. Your products fit in Jack's active site. So products become substrates. And so if we're all grouped together in the cell membrane, we can be much more efficient. Because as I break down my stuff, my reactants that are Kelly's product, my products that become Kelly's reactants are going to build up in a high concentration. So the chances of them fitting in her active site go way up if we're located close to each other as opposed to if we're just scattered randomly. Okay? And so proteins can do all kinds of things. So you can see enzymes, they can send signals, they can transport stuff. And we will talk a lot more about signal transduction later. You should just know that they can do that. Okay? Now, so membranes can form spontaneously. If we drop some lipids into water, what do? They form a ball.
That really just forms that's a membrane?
Well, it doesn't actually form a membrane, but can you imagine if I dropped some oil into water, it would form a ball.
Yeah.
And so it would be like a solid ball of oil. But can you imagine if I wanted water on the outside and the inside?
Mm-hmm.
I'd have to form a bilayer
Yeah.
in order to keep those hydrophobic pieces all together.
Yeah.
Okay? Now, but we have to get stuff across the membrane. So we can have passive transport, no energy involved. We're moving from high concentration to low concentration. Okay? And so diffusion is just the movement from high concentration to low concentration. It does not need to be across a membrane. So, remember the glacial acetic acid that stung your nose? How did it get to your nose? Diffusion.
Also, I
Ah.
was thinking it was only liquid.
Yeah.
No. Gas molecules can diffuse, right?
Yeah, like
Yes.
Perfume, various other odors diffuse from high concentration to low concentration,
Like
right?
the mosh pit at a dance. Is
Isn't there that example?
Oh, yep, right, yep.
It's more like lipids when everybody just jumbles together.
Yeah, exactly. And so we have a concentration gradient. Now, we can get things to move across. The molecules basically ignore each other if we have different types of molecules. They move against their own concentration gradient, not the concentration gradients of others. So, passive transport is movement down the concentration gradient across a membrane. And sometimes we have cells, or we have proteins that facilitate that, like channel proteins. Osmosis, now here's the gig. Osmosis is the movement of water across a selectively permeable membrane. And to get the full credit for osmosis, you have to say it's the movement of water, and you have to say selectively permeable membrane.
Can you say semi?
Semi-permeable membrane or selective, yeah, either one. Okay? And so it's the movement of water. So, concentration gradient, where there's the most free water, it moves from where there's the most free water to where there's the least free water. And so, if you look at the figure at the top of page 77, you see that we can talk about different tonicities of solution. Isotonic means there's no net movement, but is stuff still moving?
Yes.
Absolutely. So water's moving in and out,
No net movement.
but at equal rates. So if 2 water molecules come in and 2 water molecules move out, we have no net movement of water molecules. If the solution is hypotonic, and the way I remember it is you have to remember that it's the solution. If it's hypotonic, hypo with an O, the animal cell will swell up like an O and potentially burst if water keeps coming in.
Wait, if the
Solution is hypotonic.
wouldn't it be if the solution is hypertonic though, because then there's more outside, so it would go inside?
No,
Yeah, that, I'm always confused with
it's all confused. You need to think about the solution. If the solution is hypotonic, there is more water on the outside. Oh, outside.
So why would it grow?
Because
Oh,
there's
because
more
less
water.
solution and more water. There's more
So
solution
then
and
it's
less
gonna
water
go in the
inside.
cell.
Right, so water is going to go
Oh.
into the cell.
So the
Right.
net movement would be, in a hypotonic, the net movement is.
In. So in a hypotonic solution, the net movement is into the cell because there's more free water on the outside than on the inside. And so if you're an animal cell, the cell is going to swell up and probably explode. If you're a plant cell, it swells up, but it can't explode because it has a cell wall. So we call that cell turgid.
When you explode dialysis tubing, like if you don't leave enough room and the water goes in,
Well, the dialysis tubing is sort of an artificial membrane, so it actually is not going to explode. So what's going to happen is you build up, so now we're actually talking about the water potential equation. So water wants to move in, but as it moves in, you get more and more stuff inside that dialysis tubing. It swells up, and it builds up pressure inside.
yeah.
And so eventually there's so much pressure inside, it pushes water molecules back out, even though they want to get in.
So what about in an actual cell though? Is it gonna explode or is it just gonna kind of start leaking?
If you put red blood cells, human red blood cells, in pure water, they would explode.
explode or
Explode or just like
Well-
crack and
I mean,
make
it's
out?
They would lyse. Okay, they're not going to blow up, kaboom. But the cell membrane would actually rupture,
Okay.
And they would be completely.
Do you-
and then they would be non-functional. They would be dead.
They still be red.
Yeah, because they still have hemoglobin proteins in them.
And then if you put a plant cell in, the water pressure would continue to build up, and then when the pressure was really great, would the water start? Is that when the water would start moving
Right.
back
Well,
out
the
of
water
cell?
can't move in because there's too much pressure on the inside.
So the water would just start moving back out, or would there be no movement of water? Would it stay?
It's sort of there's no net movement, but we're not isotonic because the concentration isn't equal on both sides. And so then in a hypertonic solution, There's more free water on the inside of the cell than on the outside of the cell. So water is going to exit the cell. The cell is going to shrivel up.
With more free water on inside or outside?
More free water on the inside of
But
the
it's gonna
cell.
leave, so then
That's not gonna, wait, if there's more free water on the inside, wouldn't...
Water's going to move from high, osmosis is the movement of water from high concentration to low concentration.
Does hypotonic and isotonic involve just the water movement?
Yes, osmosis is the movement of water
Because
across a semi-permeable membrane.
I thought hypotonic and isotonic just involved like the concentration of solids.
Well, it does. So if I put a dialysis bag full of sugar water into a beaker of pure water, where is the water concentration the greatest?
The free water.
Outside,
Yeah.
because the inside has less free water because it also has sugar in there. And so the water is going to move from high concentration outside to inside. That solution is hypotonic. If the solution is hypertonic, imagine putting a dialysis tubing that I filled with pure water into a beaker of salt water. Where's the free water concentration the greatest? Inside. So the water is going to move from high concentration to low concentration. It's going to move out of the cell. And the cell will shrivel up. A plant cell becomes flaccid. If it happens too much and the plasma membrane separates from the cell wall, that's called plasmolysis, and that's it for the cell. It's done. You cannot revive your coleus. It stays wilted and dies. Well, then plasmolysis didn't occur. It just wilted. It just became, the cells just became flaccid, but plasmolysis didn't
When
occur.
the color changes, like in the plant, does that like indicate does that have any indication whether the cell like completely died?
Not necessarily. Well, if the cell turns brown and brittle, then the plant probably died. But if it just changes color, no, that has to do with production of pigment, and often plants produce more pigment, more purple pigments when they're stressed. Yes.
So the people that came in with the really dark coleus.
Yes, or sometimes it's a response to light. So pigment response is a response to lots of things because pigments are involved in various metabolic processes as well as protection. Okay, but that's a topic for later. Okay, the last thing I want to talk about here is facilitated diffusion, which is different than just passive. In facilitated diffusion, we're actually using a transport protein or a channel protein. It's providing a little passageway. One classic example of that is an aquaporin, which lets water molecules through, and only water molecules, and that's how we can get water in and out of cells so quickly, which is really important. Okay? If we're going against a concentration gradient in section 5.8, we have to use energy. ATP energy. And so we have an active transport protein. And notice that when the ATP binds to the molecule, it phosphorylates it, and it actually changes the shape of that molecule. And so you can see it's pumping things across. We use active transport all the time to pump things and create concentration gradients. Okay? At a much larger scale, the whole membrane can get involved, not just a protein.
Like growing? Or?
The whole membrane can get involved in engulfing something, which is endocytosis. Right, and that's when we were out in the pod, and we were wearing our little flags, and we engulfed things, and we spit things out. So endocytosis is engulfing. We can have phagocytosis, which is cell eating, or pinocytosis, which is cell drinking. And notice that down at the bottom there is also receptor-mediated, where endocytosis only happens if the molecules land on those receptors on the outside of the membrane. Exocytosis is just the opposite. We're taking a vesicle maybe full of a protein product that we made, and we're getting rid of it. We're sending it outside the cell. And that, my friends, is all there is for Chapter 5. Hi there, I'm Mrs. Gay. We're still on Chapter 4, looking at Section 4.3. Right? So, as a reminder globally, not just for this test, in this textbook, prokaryotic cells, information about bacteria, is sprinkled throughout the book. And if you remember, when we were sort of talking about this section, I encouraged you to keep somewhere in your binder a little running list of sections in your book that deal with prokaryotes. So if you haven't started one, this would be an opportune time. And so section 4.3 is really our first section. So it's reminding you that bacteria and archaea are the two domains that are prokaryotic. The other domain, eukarya, consists of all eukaryotic cells, and that includes the kingdoms protist, fungi, plant, and animals. Okay? Now, all cells, regardless of whether you're prokaryotic or eukaryotic, have a plasma membrane because you need boundaries. You need to keep the insides different than the outsides. So regardless of your cell type, prokaryotic or eukaryotic, you have a plasma membrane. You also have to have chromosomes because you need information.
So
and all cells have chromosomes, regardless of whether they're prokaryotic or eukaryotic, because they need directions to make things like proteins. Okay? No directions, no exchange of information. That cell's pretty worthless. So all cells are going to have chromosomes. The other thing that all cells are going to have are ribosomes. Okay? Why do you need a ribosome?
Make protein.
Do you need to make proteins from the information contained in the chromosome? Okay, so you're going to have those kinds of things. Now, the difference is if you are, I'm thinking about chromosomes again, if you are prokaryotic, you get one large circular chromosome. You may also have some smaller bits of looped DNA that are called plasmids, which we'll get to in a few chapters. So that's sort of a preview of coming attractions. Okay? If you are eukaryotic, your chromosomes are linear. And if you're eukaryotic, your chromosomes are contained in a membrane-bound nucleus, which is the primary distinguish.
Wait, you mean
Eukaryotic. Did I say prokaryotic?
No.
Oh, I thought.
Okay. So eukaryotic cells, linear chromosomes, enclosed in a membrane-bound nucleus. All cells, again, have cytoplasm. Now, in a eukaryotic cell, we don't refer to cytoplasm inside the nucleus. We consider cytoplasm the area from the nucleus to the plasma membrane that houses all the other organelles. Okay? Eukaryotic cells are bigger than prokaryotic cells. You saw in the diagram in 4.2. Okay? Prokaryotic cells are only about 1/10 as big as a eukaryotic cell. Okay? And so if we look at this section, prokaryotic cells also have a few special distinguishing features. Okay? Many of them have, so we'll start in the center, you see the nucleus. There's also a nucleoid. The nucleoid is not actually a thing, but rather it's a region. It's the part of the cell where the DNA is located, but it's not enclosed by a membrane. There's ribosomes near the chromosome for making proteins, the plasma membrane, of course. Most bacteria also have a cell wall. Now, the cell wall is made out of peptidoglycan, which is different than the cellulose that makes up plant cell walls, or the chitin that makes up the cell walls of fungi. So many different kingdoms have cell walls, but they're made out of different things. This is why antibiotics kill bacteria, but not us, and not plants. Because what it does is it targets the peptidoglycan, blows holes in it, and then the cell leaks.
Should
Okay?
we know what that cell wall is made up of? Should we know the peptidoglycan?
Again, that's sort of one of those things you should be familiar with, recognize, but not necessarily memorize.
Okay, can you say it one more time?
Peptido, which sounds like peptide.
Okay.
Glycan, which sounds like sugar, right? Okay. Now, we also have many prokaryotes. They have their plasma membrane on the inside. They have a cell wall. Many of them also have a capsule, which is like a jelly-like coating. It helps them stick to things, which is particularly important if you're in a liquid-type environment. Okay. Many of them have flagella, which helps them move through their environment. They also may have pili, tiny little hair-like structures on the outside that help them attach. Okay, but we'll talk, and then, so that's sort of like the introduction to prokaryotes. When we get ultimately to chapter 16, we'll talk about them a lot more. Okay. In section 4.4 on page 56 and page 57, we are going to spend the rest of this chapter considering eukaryotic cells and looking at the organelles. In this case, they're introducing you to the main differences between plant and animal cells. And so, if you look at the animal cell, they have lysosomes and centrioles.
Plants don't.
And plants do not. Well, a few plants do, but most plants do not. Okay? And if you look over on page 57, you'll notice that there's, on the left-hand side of the diagram, you can see that there are some organelles that are in plant cells that are not in animal cells. That large central vacuole, chloroplasts, cell walls, and plasmodesmata.
Are there any? There's no vacuoles in animal cells, though, right?
They're not called vacuoles. We tend to call them vesicles if they're in animal cells.
Or are they the same thing?
They can be used for storage. Notice that they both have peroxisomes. Notice that they both have mitochondria. That's key. They both have Golgi apparatus. They both have ribosomes. They both have endoplasmic reticulum, smooth and rough. They both have a nucleus, which is composed of the nuclear envelope, the chromosomes, the nucleolus. And they both have cytoskeletons made out of microtubules, intermediate filaments, and microfilaments. Okay? So, just understand maybe some of the basic differences. They might give you a list of organelles. I could imagine you seeing a list of 5 different organelles, A through E or A through D.
Which one is not in it?
And of that list, they might have 3 or 4 questions associated. So your answer choices, I'm totally making this up. A, nucleus. B, mitochondria. C, vacuole. And C, centriole. And question 2 might be found in animal cells but not in plant cells. Question 3 might be, the produces energy for the cell. And so then you would choose from that, you would use that same set of answer prompts over and over and over again. Answers could be used once, more than once, or not at all. So I could see a listing like that that would ask you to know the differences between plant and animal cells. Does that make sense? Okay. So I'm going to stop this recording now, and I'll answer questions, and then we'll carry on with the next one.